know something about a protein? First stop is UniProt, and as someone who studies proteins, well, I use it quite a lot. So I thought I would walk you through a UniProt database entry.
This isn't like a full tutorial or anything. I just want to show you the basic anatomy of a UniProt post and what type of information you can find there. And then the best way to get used to it is really just to use it. But say there's a protein you want to know something about. So the protein that I studied in grad school was called AGO2. And you can learn a lot more about it in other posts. It's a protein that's involved with RNA interference. But anyway, if I want to find more about the protein, I can search for it in the search bar. I could also go straight, if I had the accession number, which I'll mention in a second, I could also use that to find it. And so you can see that it'll show you things that are related to your search term. This is the one that I want to look at. You can see that it has been curated. So there's this reviewed status, so this gold thing. Basically, UniProt consists of, it has both like a reviewed part, which is like this Swiss prot side of things, and then the unreviewed. So it's like Tremble. So there's a lot of sequencing data and that sort of stuff, as well as predicted sequences based on species having really similar looking things. So anyway, there's a lot of annotation that's needed to make a really nice high quality entry, make sure that everything has been everything is legit, all of that sort of thing. And so there's sequences in there in the Trumbull side of things that aren't that validated, verified, gold standard type of thing. And so sometimes you'll have to work with those, but this gold standard tells us that this has been well annotated, and you can see it has an annotation score of 505, 5 out of 5. So basically, when you talk about annotation, it's just like putting information, like curated information associated with a gene or a protein or something like that. And there's all sorts of different information that can be associated with a protein. So just in terms of the entry name, so this is the entry name and this is the accession code. So this is going to be like a stable thing that you can then enter this into different databases, or not different, yeah, different databases or different software, and use this to look up the protein. And then it also have an entry name. So this is one that makes it easier to actually like know what they're talking about. And so, but this could change, but this is never going to change, is the basics. But it'll have the gene, like a code for the gene, like the gene name, and then an underscore, and then the species. It'll also have different information. So this is an enzyme, so it has an enzyme commission number, and it'll tell you the length, and that this has been well annotated. And then it has all of these different features that you can look through. And so we'll go through all of these. So it starts with the function. What does this protein do? If it's an enzyme, it'll have information about its catalytic activity. So how it does whatever, how it speeds up the reactions it speeds up, even kinetic information, on some structural features. You can also jump to different things you want based on here. And so often the reason why I'm going to a UniProt entry is to try to get the sequence. Like the protein sequence, you can get directly from UniProt, and then it'll give you links where you can go and you can find the mRNA or the DNA if you need that for other purposes, and you can find it's like genome location, all of this various things. And so one of the things I'm starting with the sequence and isoform both because it's what I typically am using UniProt for often, as well as because it can be confusing if you're not exactly sure what you're looking at because of this isoform. And so basically there can be different versions of proteins that can be made based on using alternative translation alternative slice sites, different translation start sites. So various reasons why you could have different isoforms. And so you can see that here you have this canonical sequence, and then this is saying that there's another version from alternative sequences splicing. So this is the one that's chosen as canonical. So this is like the one that people are usually referring to. And then there's this Q and whatever this 2-2. So this, remember, this is the accession number, and then you have the dashes for the different isoforms. And you can see that this one, it tells you that it's differing from this one and that it's missing this region. And so sometimes it's not always that the canonical one is like the most common or that sort of thing. I'm not exactly sure how they determine which one is canonical, because sometimes the different isoform could be actually the one that you're looking for. So don't just because it's non-canonical be like, oh, that's not the one I want, but it'll likely be the canonical one that you do want. So you can see that it'll tell you the length in the terms of the number of amino acids, as well as the mass in Daltons. So typically, a lot of times when we're talking about proteins, we're talking in terms of kilodaltons. A kilodalton is 1,000 Daltons, and so that would be 97 kilodaltons. Or 97 kDa. And you can see that this one is about 94 kDa because it's missing that region. So this is why you could potentially have different isoforms. And if you wanted, you can align them. And you can also align to different proteins. What you can do is there's this tool called, or there's this add to basket, this like little shopping thing. You can add it to your basket, and then it'll have sequences in your basket that then you can click and you can compare them. It has this BLAST tool. So we talked about BLAST before, how we could use it for nucleotide sequences. We could also BLAST protein sequences, perform alignments. Things like clusteral omega is really good for that it's hooked up to, will allow you to compare sequences and align them. So you can also find the information about, if you go down to the bottom, like similar proteins. So this will show you based on, you can choose the amount of similarity you want. So here I'm getting proteins from, if I go down in similarity, I'm getting proteins from krill, and I'm sure what it is, horse, olive, bovine. And if I want 100% activity, then you can see it's just like identical to the chimpanzee's protein sequence. And then if you wanted, you could blast them and compare them. Okay, so that's the protein sequence. And so you can see that when we look at these letters, we're looking at the protein with the amino acids, with the 1-letter codes for the amino acids. And if we wanted, we can download these, and we could also copy the sequence. And so you can see that when you click on it to download, it's in this format that is this conventional like FASTA format that is used for protein sequences, as well as like nucleotide sequences and this sort of thing. Using the same format makes it easy for software to then like parse or read in the file and see which part's the sequence and which part is like the name and that sort of thing. There are also, we talked about the different tools. And so you can compute the PI, the molecular weight. So the PI is like the isoelectric point. So if you want to figure out like if it was charged or at a certain pH. Prop param is, it'll link you to prop param, XKZ prop param, which I've talked about before, which is really great for getting information about like the PI, as well as the sequence composition, number of different amino acids, the extinction coefficient for getting the concentration from UV, etc. There also, so it also has, those are the 2 isoforms that have been like annotated, but then it also has like computationally mapped ones, which you should treat with caution, but they're potentially, these have been found in like sequencing studies or based on like comparison to other species. There's various reasons why you would have computationally mapped ones, and you can, if you press on this eye, you can like get more information about all of these different things. Okay, it'll also show you like sequence features. So there could be in the references in different sequence databases, they might have differences from the canonical sequence. And so it'll tell you more about this. So where there's like sequence conflict, and then you can see where that is. Okay, so that was the protein sequence. And if we want to, you can also like zoom in various things, lots of different stuff. Okay, so that's the protein sequence, but what if you want to know where it's located in the genome? Or what if you want to know where it is, like the DNA sequence, the coding sequence, the version of the DNA that's actually used to make the protein. So basically the cDNA, the complementary DNA of the messenger RNA. Because the DNA is going to have a bunch of exons, and those are going to get spliced together to give you your mRNA. That is then, or I mean, like the messenger RNA copy the DNA is going to get transcribed, then the codons are going to get spliced out to give you remove the introns, so the regulatory regions, and splice together the exons, so the protein coding parts. And so if you want, you can then look and you can go and see where in the genome it is in this genome annotation database. If you want to know like the coding DNA sequence, so you can see, remember here, there's those two isoforms. And so here we're seeing the -1 and the -2. We said that the -1 was the canonical one, so let's go and look at that. This link is going to take us to the CCDS, which is the consensus CDS protein set. So coding the coding DNA, and so coding for proteins. And so basically, it's showing us the coding DNA sequence for the consensus ones. This has been well annotated, curated, verified. What's cool is that, so it'll show you so this is like the nucleotide sequence that's coding for the protein. So you can see it starts with ATG. That's our start codon. And actually, if you click on it, you can actually see in the translation that it shows that that's going to be this first methionine. And so what's a really cool tool here is that you can actually click on the different codons and see where they are in the protein. This can be helpful if you're trying to, say, make a truncation of a protein to study in the lab. You can also see this like blue and black. These are showing you the alternating exons, so where the splicing together happened. So this is a cool tool, and it'll also tell you more about the chromosomal location if you wanted to then go and look and see where it's actually located in the genome. Okay, so I guess I just took you to, so you'd have to then search into that. But yeah. Oh, here it's showing you the chromosome start and stop locations, and so you can see that because there's all those exons, it'll be a lot longer, and there'll be introns, and so it's showing you where those all start and stop and stuff. So cool stuff is that you wanted to look at the nucleotide level stuff. Okay, but let's get back to the protein. So let's go to the very beginning. So what does this protein do? So it's involved with RNA interference. So basically, it takes these short little RNA pieces that are about 22 nucleotides long, and it uses these as guides and goes and seeks out target messenger RNAs that have a complementary sequence, and then it binds to them and it represses them. So it's this post-transcriptional regulatory method. And it's really cool because it's using this small RNA as a guide, so like a microRNA or an siRNA. So it's sequence directed to specific messenger RNAs for degradation, and it's really cool. And I'm going to stop myself from going too far into it because I could talk about it for days because it's what I studied in grad school. But anyway, so if you want to learn more about that, I have way more on my blog and channel and everything. Okay, but so like as I said before, it's an enzyme, so it can actually cut the messenger RNA when it binds to it, and so it has this catalytic activity. And so it has this enzyme commission number. I've talked more about these before, as well as it uses a metal cofactor. And so you can have information about that as well. And if you wanted to see like, okay, well, where's the source of this? Like how did this annotation come to be? It'll tell you where a source that you can find the like proof. And so this is one of those aspects of annotation where this is like the curated part where people go through and put this information that goes along with that gene name or the protein name. Okay, you can also get kinetic information, etc. Okay, then it tells you about the features of the protein. So as we saw before, down below, it's about like 900 amino acids long, and different regions of the protein have been found to do different things. And so you can find out more about what different regions have been found to do, as well as the annotations for domains, so different like regions of a protein that have given have different functions, or they're structurally they have a structure that can fold on itself. That'd be like a structural domain. But the basic thing is it's kind of like rooms in a house. So your protein will be your house, and then you have different domains where you would have different rooms, which could be kind of like structural domains, or you could have like different parts in the same room. So maybe you're living in a really small, like efficiency apartment like me. And so basically your bedroom and your kitchen are in the same room, but they're separate, they're functionally separated. Um, you can also have regions that where they show interaction, various things like this, and you can find out more about all of these things. And it's very nice. Okay, so now more about function. This thing called Go annotations. So Go stands for gene ontology, and it's basically a type of annotation that tries to put function with different molecules. So with genes, with proteins, etc. And so people will go through and, or they'll be computationally um, predicted and say, okay, this protein is associated with this activity, it's located here, blah, blah, blah. And then they put all of these like terms that go with the entry for it. And these terms can be grouped into different categories. And this allows you to compare between a lot of different genes and proteins to say which ones might have similar things, or if you do some sort of like high throughput assay. So something where you have like, you end up getting a lot of information like about tons and tons of different proteins, and you want to say, okay, well, what does this protein, like what type of proteins are enriched? So if you knock out a gene, so you remove one gene product, what happens to the rest of them? Do you see that like, oh, all of the ones involved with, I don't know, with cytokinesis, those ones are get affected or that sort of thing. And so basically this is just a way to organize all of that. And so you can also see, okay, like what if you do some sort of assays or some sort of experiment and you get a hit. So like you find that when you do this experimental thing, this one protein pops out of whatever experiment you're doing, and you don't know anything about it. So you would look and see, okay, what's this Go terms enrichment? Okay, so this protein, it's involved in RNA binding and regulation of DNA templated transcription and mRNA metabolic processes, or gene silencing by RNA. So you can find out information about a protein based on this. Go annotations, they'll be like grouped into like molecular function, biological processes, as well as like cellular component, like where it's located. And then you can find more about these in all of these different links. So again, as I was talking about yesterday in terms of databases, these like UniProt is one of the like launch pads that'll take you to a bunch of different resources. So you can, if you want to know more about like the pathways it's involved in, well, there are other databases that this can link you to. You can also, if you were in those databases, you could use start from there and then use the UniProt accession code that this thing that we saw at the beginning to find it. Another thing is that proteins will often have multiple names. Often this is because when people discovered it, they didn't know what it was, and so they named it one thing and then discovered that it didn't actually do that, and then so people started calling it a different thing. Or there might be that multiple people found the same protein and they were referring to it with different names, and then they all, oh, we're studying the same thing. So there are different reasons why proteins could have different names, and so it's helpful that UniProt lists them all so that you can know if you're reading, like maybe you're reading an old paper and it's referring to it by another name, and then you can say, oh, that's the same thing as here. So yeah, so you can also see if like different gene names. And so this is again that when the annotation, so I mean like, sorry, with the session number, that's going to be stable, but with like the gene name, the part that's going in the entry name, that could change, although it doesn't do so commonly, I don't think. But you can also see that it's had there are some secondary accessions. So basically other proteins in the past that were assigned to this, and then were all found to be the same thing. And so we're merged into this single entry with this primary accession. But if you were to search for one of these, you would still be able to find the updated accession number. But this is just merging these all into one, not changing that primary accession number. And so this is why it's really important that you have this one stable number that you can then look in a bunch of different databases and see that you're studying the same thing if a different one was used. Okay, there's also other bunch of other databases you can go to. It'll show you where it's located subcellularly. So there's some of it is in nucleus, some of it's in these P bodies, in the cytoplasm. I'm not exactly sure how it determines where things are. Yeah, but I'll tell you more information. Okay, then I'll tell you information about variants and disease variants. So sometimes there are mutations that cause like amino acid changes or different isoforms to be made, and these can affect the functioning of the protein. Sometimes they can cause disease. Sometimes you don't know if they cause disease, but you would predict that they would because they make some huge radical change to the protein. But sometimes there are like variants or versions that are known to be associated with different diseases, and UniProt will tell you information about those. And you can find the different mutations in the protein that have been linked to a certain disease, as well as the evidence for them. Again, it's really important that it's showing you the publications, then you can go analyze those publications yourself. It'll also show you on the sequence level where these changes are located, where mutations are located, as well as what effects they have on the protein, if it's known. Then there are variants. So variants are often like not necessarily like disease causing or anything. They could just be changes in the protein that have been observed. And so you could have like changes that I'm pretty sure that you could have for mutagenesis and stuff. This could be like experimentally determined. So yeah, so I think that this was basically like people did this in the lab a lot of their times. And so then you can test the function of different regions. And so people do that in the lab, and then the information will be put here. But then there are also variants that have been found to actually happen in these proteins, like in people and things. And so some of these variations, we don't know what happens, like what the result is, because there's none of that like experimental evidence to go with it. But we know it is present in some people. Some changes are probably not a big deal, but others could be a big deal. And so you can see that some of these are predicted to likely cause disease, and some of them are likely not, et cetera, et cetera. And so you can then see the various ones that have been detected, where they've been detected, the evidence, everything like that. Organism-specific databases, genetic variation databases. So again, you have so many sequences. That's why we have that like one consensus sequence and everything, but there could be different versions, especially if we're talking about at the DNA and the RNA levels rather than at the protein levels. There can also be differences with processing. Proteins can get like chopped up afterwards. So you could have like peptidases or proteases actually cleave the protein, remove a part of it, maybe like a signal peptide, or that sort of thing. You could also have post-translational modifications like phosphorylation, glycosylation, so adding phosphate or sugar groups, various things like that. And so you can see here that there's this cluster of phosphorylation sites that I actually studied in grad school. So you can see these modifications here. And what else? You can also get things about other types of modification. And then you can get information about where it's expressed. So I mentioned yesterday this gene expression databases, like this expression atlas, and so you can see here where it's expressed. Also, if you want to know what it interacts with, so they have this chart where you can then see, okay, this protein, it interacts with this, it interacts with this. You can filter them in various ways, see the evidence, all of that cool stuff, as well as protein-protein interaction databases. Okay, we're almost there, people. Okay, structure. So you can see that there's a lot of different structures of this protein. Some of these are going to be the full and some of these are not full. So it's kind of confusing when you see this because the first one it's showing you is actually just a part of the protein. It's just one of the protein's domains. And so you can see here that it's only positions 439 to 575. If you want to one the whole protein, then you go down and you can see, okay, this one. So this is the 4F3T, that's its PDB code, so you can find it in the protein data bank. This is a protein that was solved by a lot of time in the lab of Lord Joshua Toro, where I did my PhD. And so you can see here, now you have the full protein. So don't be confused. A lot of times it'll have one of the older structures, the earliest solved structures, or maybe it'll be a just a domain, just a part of the protein, and then later you'll get a structure of the whole thing. For various reasons, that it's a lot easier often to get a structure of just a part of it, and so then it takes a lot of not that people in the beginning didn't have to work really hard to get that first part, but then once you do that, then there's better technology. You can build off of the first results, end up getting a full structure. And then, so based on that structure, you can get information about which regions of the protein have loops, which have helices. So those like curlicues, which have turns, which have beta strands, then beta pleated sheets, and et cetera, et cetera. So those various positions. And then you can even just look at that part of the protein, align it to see what other proteins in the data bank have a similar structure. And this can, because different regions of proteins might, or different, like because of evolution and things, different proteins might have similar regions, and you can derive from the same source and have a similar function. There's also like the AlphaFold prediction that it gives you as well, and so you'll see a structure even if there's no solved structures of the protein. You will see like a predicted structure, and so this you'll also see that the predicting structure, so it'll show you like the confidence of various regions. So you can see that there's some regions that are very unconfident, like this orange region, and a lot of times these regions are actually unresolved in the crystal structure. So even though they're present in the crystal structures, you can't see it, and so these will be the regions that in the protein structures will be shown as like those dotted lines, those dashed lines, or if it's too big, it won't even be there at all. But you can see these dashed lines, those are like regions of the protein that the AlphaFold will show you, and they'll often be in low confidence because that's just the predicted structure. Finally, there are things like, where was we? Yeah, so we talked a little bit before about different domains of the protein, like the rooms in your house or the functional parts of your house. And then there are various databases that are based like around domains, as well as you can get like evolutionary information like phylogenomics. So there's links to all of these various things. And again, if you were to start in one of those other databases, you could then use the UniProt accession code to find this information. And then we are back at our sequences. And so we did it. We went through the whole entry. And so you can see that there's just a lot of information here. And it's not even like, I just went briefly over it. I was hoping to be briefer, but I wasn't. But there's just so much there. And I wanted to just show you what was there. And so I hope that this will help you. Really, the best way is to just go and explore. There's a lot of other information on the UniProt, as well as like tutorials and training. If you are unsure of how to use it, you can find like a full tutorial as well. And so yes, so huge thanks to everyone at UniProt for making this great free resource. It's totally free, and just a great source of information for researchers around the world. And so I hope it can help you.